So the spotlight feature is on. Please turn on your microphone and video, and let's start with today's class. Just give me 1 minute. I will share the screen with you. Yeah, can you see the screen? Okay, great.
Yes, sir.
Hi.
Hi.
Thank you so much. Okay, how are you doing, Hiram? All good?
Yeah, good. Yes, sir. Good.
Perfect. Okay, so let's start with just give me 1 second. Let me see if others are joining. I gave you homework in the last class to go back and look at spermatogenesis, and also whatever we have done till now, we have to make notes, right? Okay, perfect. So did you find any problem with respect to the anatomy of the male reproductive system, the organs, glands, anything?
No, not yet.
No? Okay, perfect. So let's start with a quick revision, quick summary by you of mitosis versus meiosis, what we discussed, and how is the male reproductive system regulated. Yeah.
Yes, sir.
Okay, great.
Okay. So in the last class we discussed with mitosis versus meiosis. Mitosis is equational division, whereas meiosis is reductional division. In mitosis, ploidy remains the same, and in meiosis, the ploidy is halved. Mitosis results in the production of 2 daughter cells, and meiosis with 4 daughter cells. The 2 daughter cells are diploid, and here the 4 daughter cells are haploid. Mitosis happens in somatic cells, whereas meiosis occurs in sex cells.
We also learned how to define haploid and diploid, right?
Yeah,
You remember
haploid
that?
is, yes. Haploid is n, or half the number of chromosomes, whereas diploid is 2n, a set of chromosomes.
Yeah, but when you say half the number of chromosomes, what is the full number then?
Full number is
Remember
40.
46.
Yeah.
But it's for humans. Remember that?
Yes.
I give you an example that in some other organism, like let's say a fly, fruit fly, the full number is 8.
Yeah. It is.
Right?
Yeah.
So there was a better way to define haploidy and diploidy, apart from just saying that it is half the number of chromosomes or full the number of chromosomes. What was do you remember that? Did you note that down? So haploid means one copy of
One
the
copy
genome.
of genome. Yeah.
So if you say, or if you define that haploid cells or haploid organisms are those cells or organisms that contain just a single copy of their genome, then it's haploid. Now, by saying this, you are taking out the problem of defining what is the copy. Whatever is the copy, whether their full genome contains 8 chromosomes or 10 chromosomes or 46 chromosomes, does not matter. Any organism who just have one copy of the genome is haploid, or any cell that has the one copy of the entire genome is haploid. And if a cell or an organism has 2 copies of the entire genome, in terms of chromosomes, of course, so there will be a same chromosome in 2 copies. Like chromosome 1, it will be having 2 copies of chromosome 1, one coming from the mother and one coming from the father. Then that cell will be called as diploid. So a better way to define, you can write, both of you. Rabia has joined in. Hello, Rabia, how are you?
Fine, sir. How are you?
I'm good. I'm doing good. My voice is clear to you? Clearly
Yeah.
audible and visible? Okay, great. Yes, so we were discussing that the good way to define haploid and diploid is that a haploid organism or cell contains one copy of its entire genome, just one copy, versus a diploid organism or a diploid cell contains 2 copies of the entire genome. So each chromosome has a homologous another chromosome in a diploid cell. Okay, We will learn more about homologous chromosomes in genetics, but I'm priming you. I'm making your foundations for genetics right here. Okay. Yes, yes, Iram. So let's continue with how is the male reproductive system regulated. Summary.
Yes, sir. So then we discussed how is the male reproductive system regulated. Uh, just wait, sir.
Mhm.
Yeah.
Yeah.
Yeah. So it starts with hypothalamus. The hormones produced by hypothalamus are stored in the pituitary and
Not stored. They are just so hormones are not stored anywhere. They are like messengers. So they are secreted and they go and act or they bind somewhere to give the message. Okay. But the important thing that you are saying, the important thing is that hypothalamus only secretes two kind of hormones. Whatever hormone it is secreting, it is either to give a excitatory signal to pituitary, activatory signal, or a inhibitory signal. So in this case, hypothalamus secretes GnRH, activatory signal to the pituitary. It goes and bind to pituitary, not it doesn't. It's not stored there. Okay. Yeah. Continue.
So hypothalamus secretes GnRH, gonadotropin hormones, and it goes and acts on pituitary, and the hormone binds on gonads, and this leads to the formation of sperms. There are two types of gonadotropin, FSH and LH. FSH will initiate spermatogenesis, and LH will secrete testosterone, and this will maintain spermatogenesis. And here a question comes that which hormone is responsible for maintaining spermatogenesis at any time? The answer is testosterone.
Yeah, but to initiate it is
It
FSH.
is, yes. So this is the time where the male reaches puberty, and testosterone also induces a secondary sexual character in male. Example: deep voice, facial hair and pubic hair, and growth of muscle. Also, sexual drive, and sexual drive will lead to erection of penis and ejaculation.
Yes, right. Perfect. So this is where we finished in the last class. In today's class, we will just give me 1 second. Yes, we will begin with spermatogenesis in detail, this whole concept in detail. So let me just pull it a little up and make sure that you can see it. Okay. Let me know if it's visible clearly. Too much. Yes, is this visible? Visible clearly? Clearly. Like and it explains things in taken it Britannica Encyclopedia. And before discussed, yeah, let me take you through this diagram first, then we will discuss the concepts and important. So this part, spermatogenesis and oogenesis, is very important for your boards and entrances, both as many questions as you want until this whole process is clear. Okay, so the board kind of looks very busy, but let's try to make it easier. So let's go one thing. So first let's look at, you can see my cursor, right? This pointed cursor that's hovering. Can you see that? Okay. So let's look at this structure, which is testis. Okay. And as I told you, inside the testes, there are chambers, right? Lobules. So can you see, if I draw it for you, you can see chamber like structures, right? Can you see the chambers? Yeah. And these are called testicular lobules. And inside the testicular lobules, there are these small ducts called seminiferous tubules. So they have taken one seminiferous tubule out and they have magnified it for you to explain things. it is inside these seminiferous tubules, which are present inside the testicular lobules, where spermatogenesis takes place. And to just put things in context and to just revise what we have discussed already with this structure, just a little bit zoomed in here, you can see, can you see this network-like structure formed here?
Yes, sir.
Can you tell me what is this then? If you have understood from the previous, when all these seminiferous tubules come and form a network-like structure, what do we call that part? Remember, there was something that I told you, we call it retetestis, right? So you can write down all seminiferous tubules from all the testicular lobules come together and converge towards the center of the testicle, or towards one side, you can say, which is called retetestis. So retetestis forms a network-like structure. Okay. And if I highlight the structure that I'm highlighting, can you see the highlighter? The yellow highlight?
Yes, sir.
The structure that is being highlighted, what is this? That goes out of the retetestis, and it's highly convoluted, coiled, and then it connects the testes with the epididymis. I told you in the last class. What connects testes with the epididymis? These are all questions, by the way, board questions. So entrance question. So perfect. It's called vasa efferentia. Okay. Vasa efferentia. Then you can see this is the testis, a comma-like structure, right? Sitting at the top. Sorry, this is the epididymis, a comma-like structure sitting at the top of the testis. And what is epididymis? What is it famous for, or what is it known as? Anything special about it?
Functional maturity.
Yes, it is a site of functional maturity for sperms. So I think you can now make a mind map and understand, if sperms are being formed in the seminiferous tubules, they travel through the seminiferous tubules, go into the retetestis. From the retetestis, they go into the epididymis through the vasa efferentia. And in the vasa efferentia, it's like a camp for them where they stay for some time, around 2 weeks, and they get functional maturity. Correct? So this is the part where they enter. Let me use a green highlighter here. So this is the part where they enter in the epididymis, and then they travel all the way through epididymis and reach this end. Okay, here. And from here, then they start going up. And can you tell me what is this duct which is going up from the epididymis? Yes, this is vas deferens. Perfect. So is the overall anatomy clear to both of you, Rabia and Iram? It's very important.
Yes,
if you understand this anatomy, many of questions becomes easier. Now, what I want you to do is, we will just focus now on a very, very niche region, which is this seminiferous tubule, which is zoomed in for here. So you see, let me use red here. This I have drawn the center of the tubule, right? Can you see the center? And can you also see that this is the lumen? Lumen means the empty space. So this is the lumen which I am encircling with the center. So only this much is the empty space. And from here till here, this whole thickness is the wall of the tube. Okay. So it's a very thick walled tube. It's not like the normal hose pipes that we use to water our gardens, where the membrane, the boundary is thin and lot of fluid can go through it, right? It is opposite. Very less space is there in the center, and this space in the center is called lumen, the empty space. And the wall of this tube, seminiferous tubule, is very thick. Why it is very thick? Because this wall of seminiferous tubule is the site where everything is happening. Okay, everything with respect to spermatogenesis is taking place in this wall. So what they have done here for you is very beautifully, why I like this diagram is, They have taken this cross section, which I am, and this rectangle, and this rectangle is shown here. So in a way, what you are seeing as a zoomed version of the wall. Is it clear, Iram and Rabia?
Yes, sir.
Perfect. Now let's see what's happening in the wall. So towards the top, which is here, now it is here. This is the outer layer, right? And towards the bottom, which is like this, it is lumen, right? So everything is happening between the outer wall and the lumen. Is it clear? Now, here comes the most important part that is asked. First question is, what kind of cells are present in the seminiferous tubules? So there are majorly that you have to be interested in. Two major kind of cells are important for spermatogenesis. One is these cells that you are seeing. See, the ones which I am highlighting in green here. Okay. The one which are highlighted in green are called spermatogonium cells. These are the cells which are round in shape, but over the time they will develop into sperms and will become, unlike what they are now, they will become elongated and mature sperms. So these are one type of cell. The other type of cell, which I will highlight with, let's say, blue, or let's say color. Yellow will be fine as well. Yes, is this cell. So it's little, just pay attention to what I'm trying to show you. I'm drawing the boundary of this cell and you will understand what I'm drawing. Can you see this big cell here in this cell? Yeah, this yellow part, basically the
Yes,
whole yellow
sir.
part. So what I have marked in yellow are called Sertoli cells. Okay, so these are the two kind of cells which are important here. Okay, the spermatogonium cells and the Sertoli cells. Now, spermatogonium cells, as I told you, they are the precursors of sperm. Sertoli cells are also known as, they act as nurse cells to the spermatogonia. Okay. Now they do not have a fixed shape, plus they are big cells. And write down that spermatogonium are embedded in Sertoli cells. Okay, so Sertoli cells kind of cover the spermatogonium from all the sides. Okay, so they're kind of embedded or totally surrounded, if not embedded, totally surrounded by Sertoli cells. Now, what does a nurse do in a hospital? What does a nurse do? So you'll be
Take care of the
becoming,
patients.
take care of the patients. So can you elaborate that taking care of the patients part? I know you both want to become doctors, but you should know what your fellow nurses will be doing for the patients, right? I think you know what they do. So taking care means right from helping them with the movement to healing to giving them nutrition, which is food, medicines, which is again in that context we can call it nutrition to heal, or drugs. And if patient wants to move around, if the patient has a fracture or something, patient cannot move on its own, then nurse cells are the one that help the patients move. So from locomotion to nutrition, everything is taken care by the nurse. Correct? Am I correct? So these cells, which are called Sertoli cells, also do the same thing for spermatogonium cells. They provide mobility, they provide nutrition, protection, everything to the spermatogonium cells until it becomes sperm. Okay, so everything is taken care by these cells. There are other very important cells which are present outside these tubules. So in this testes, if you pay attention here, let's say this is a seminiferous tubule that I'm drawing in red. Okay, I'm highlighting in red. So these are seminiferous tubules, let's say. But do you see there is region apart from it, which is where the seminiferous tubule is not present, like this region where the seminiferous tubule is not present, right? So what is present in this part? So outside the seminiferous tubules and inside the testes, inside that testicular lobule, this part is called interstitial tissues. Okay. Inter means between, interstitial. So between two seminiferous tubules, there is interstitial tissue. And in those interstitial tissue, a very important cell is present, which is called Leydig cells. So write down. Leydig cells are present outside the seminiferous tubules. Leydig cells are present outside the seminiferous tubules in the interstitial tissue of the testes. In the interstitial tissue of the testes. Is that clear? Okay. Now, what are these Leydig cells responsible for? So these, remember FSH, sorry, LH from pituitary. I'm just connecting dots. LH from pituitary, when it comes and signals the testes to produce testosterone, ultimately some cell inside the testes has to take up this job, right? For example, I as a postman or I as a messenger came to your house, knocked on the door and told someone in the house that there's a message from someone that this needs to be done in the house, but someone has to do it in the house, right? So that someone is Leydig cells. Leydig cells secrete testosterone. Okay, write down. Leydig cells are responsible for secretion of testosterone. Okay, testosterone is a kind of androgen. The word andro in biology means males. Okay, and you have not studied it yet, but I think in class 11th or class 10th, if you know what is an androecium and a gynoecium in flower, right?
That.
You studied that, right? So androecium is
Male reproductive part.
male reproductive organ, right, of the flower. It's divided into two parts, stamen and anther and filament. It's also called stamen. We call it androecium. Gynoecium is the which we call pistil. So the word guy, and you must be knowing doctors who are called gynecologists, right? So can you figure out now the word, what does gynecologist means? Logi means study. So logist means someone who studies that. Biologist is one who studies biology. So gynecologist is the one who studies, or who's the doctor of female reproductive system, right? Female reproductive system, which includes childbirth as well. So that's called gynoecium. Or when we come to hormones, so male hormones are called androgens. Okay. And female hormones, what will they call? Can anyone just tell me? Just simple. I told you everything. Just make a guess. What will you call a female hormone then?
Gynogens.
Of course. Perfect. We'll call them gynogens. But we mostly use the word androgens for testosterone. So testosterone is androgen. So we sometimes in entrances, they also ask you the question, which of the following cells secrete androgens? They will not use the word testosterone. They will use the word androgen. So the answer is same. Leydig cells secrete androgen, and they are present in the interstitial space. Is it clear? That's one important cell. Second is, let's come to Sertoli cell back, because we are studying the process of spermatogenesis. So Sertoli cells are present inside the seminiferous tubule. in the wall of the seminiferous tubule, not in the lumen. In the lumen, nothing is present. That's why it's lumen. So lumen is the empty space through which the mature sperm can go out into the retrotestis. Okay. So Sertoli cells are present in the walls of the walls of the seminiferous tubules. And now let's focus on this part of the diagram here. How does spermatogenesis begins? Now this is very important part. Remember meiosis? We talked about meiosis, mitosis. That's clear, right? Everyone? Rabia? Any doubt
Yes,
with meiosis
sir.
and mitosis? No doubts, right?
No.
Okay. So everything starts with this primordial germ cell, PGC. It is called very famously. And just to tell you, as a part of interest, my MSc research was based on PGCs. Okay. So PGC, but not in humans, but in a fish called zebrafish. That fish has zebra-like stripes. So PGCs or primordial germ cells are basically very early when the embryo is being formed. It is decided very early on that some cells are special and they are called primordial germ cells. And these cells in the embryo reach a specific place and stay there, and that is the region where gonads will be formed. So primordial germ cells form the whole gonad, which is testes and ovaries, and inside the gonad also the gametes. Is it clear? So write down, PGCs or primordial germ cells are responsible for the formation of gonads and gametes. PGCs. Okay. So first gonads, let's imagine gonads are now formed, okay, in the embryo. But gametes won't be formed in a male until puberty reaches, right? Before puberty, the human male produces no sperm at all. So these primordial germ cells stay in the gonad, okay? And inside the gonad, they stay like that. Once puberty reaches, they start multiplying to form spermatogonium cells, which are marked here in green and are at the top of the layer. Can you see these green cells which I have marked at the top of the layer? These are the first cells that start this process. So PGCs develop into spermatogonium cells through mitosis. This is simple mitosis. No meiosis is happening as of now. Remember I told you that inside the gonads also mitosis happens just for the development of gonads and for anything else. Meiosis very, very specifically and specially happens just for sperm production. So first step from PGC to spermatogonium, mitosis happens. Okay. So in a way, PGCs are diploid, right? To N. So what will be spermatogonium? Spermatogonium will be diploid or haploid? Tell me. Rabia. Spermatogonium cells which will form sperm later on, what do you think? They will be haploid or diploid?
Diploid.
Diploid. And why is that? Mature sperm is haploid and spermatogonium cells are forming that sperm. Just to tell you, mature cells are haploid. So what do you think spermatogonium will be? And primordial germ cells are diploid. So spermatogonium will be haploid or diploid. I'm just trying to trick you, confuse you. You said diploid. If you stick with that answer, you just have to justify. Why do you think it will be diploid? Or do you think it's not diploid, it's haploid? Rabia. Every hint to help you is on the board. Hiram, what do you think?
Haploid.
Haploid. And why do you think so?
Because sperm formation is probably haploid.
Right. So that's why I brought that sperm haploid part to just confuse you. See, the hint is this: spermatogonium is formed through mitosis. Anything which is the result of mitosis. Let's go up. The daughter cell of mitosis is diploid. Whenever there is mitosis happening, there can never be a haploid cell produced from a diploid cell, right? So to start with, PGC is diploid, then it undergoes mitosis to form spermatogonium. So spermatogonium has to be diploid, right? Is it clear?
Yes, sir.
Then these spermatogonium cells again undergo mitotic division, which is written here. Again, mitosis happens and primary spermatocyte is formed. Now, this name is very misleading, not misleading, but it's confusing because it says primary spermatocyte. The word spermato means sperm, cyte means cell. So often students think that this is the primary sperm. This is the sperm, right? They are not wrong. This is the one cell which is going to become sperm. But primary spermatocyte is also diploid. It is not haploid. Even though the name is spermatocyte, it is diploid. Why it is diploid? Again, the same reason, because spermatogonium form primary spermatocytes through mitotic divisions. Is it clear, Iram and Rabia? Yeah.
Yes,
So write down
sir.
both spermatogonium. Write down both spermatogonium and primary spermatocyte. Both spermatogonium and primary spermatocyte are diploid cells. Are diploid cells. As they are formed by mitotic division. As they are formed by mitotic division. So look at the schematic here. The schematic will help you understand few things. See primordial germ cell. Can you see this schematic first? Yes, primordial germ cell, this one. Then it's just proliferating and dividing. It is maturing and then it shows division. And how many daughter cells does it give? 1, 2. And these two daughter cells, what are they labeled as? Spermatogonium. So can you get a idea from here that this is mitosis happening? Because if it was meiosis, there would be 4 daughter cells, correct? Is it clear?
Yes,
So from
sir.
PGC to spermatogonium, it's mitosis, and hence the spermatogonium is diploid. And from spermatogonium, also they have not shown it like that, but just one they have shown. But here also it is mitosis. So from each cell there will be 2 cells formed again. So primary spermatocyte also is diploid. Now this is one entrance question from I think 2014 paper. From one PGC, how many primary spermatocytes can be formed? Write down the question. And then I'll not give you options. So Rabia, you are going to tell me the answer. From one PGC, one primordial germ cell, how many primary spermatocytes can be formed? Whenever you finish this, you can tell the answer, Rabia. This part is confusing. That's why we are spending time on it, and it's very important concept for reproduction. Yes, Rabia, what is the right answer? You there, Rabia? Am I audible to you?
Yes.
Yeah. From one PGC, how many primary spermatocyte?
Two.
Two. And what is your answer, Iram?
Four.
Yes. So the right answer is 4. Because from one primordial germ cell, there will be 2 spermatogonium. Don't forget there's a middle stage called spermatogonium. So from one PGC, 2 spermatogonium, and from each spermatogonium, 2 primary spermatocytes. Because there is mitosis happening twice. One between this stage and then between this stage, right? So ultimately, there will be 4 because there is a stage in between called spermatogonium. Okay. Is that clear, Rabia? If the question is
Yes,
from one
sir.
spermatogonium, how many spermatocytes? Then you will say 2. Okay. Okay, now let's move forward. Write down the next important point. Primary spermatocyte, write down. Primary spermatocyte is the first cell type. Primary spermatocyte is the first cell type that undergoes meiosis or meiotic division in the process of spermatogenesis. Primary spermatocyte is the first cell type that undergoes meiotic division or meiosis in the process of spermatogenesis. Okay. And can you also see, let me highlight it in red. Can you see these are primary spermatocytes which are marked here, right? These. Now, do you appreciate that they have slightly come down? They are not exactly at the boundary, they are coming down. And if you look at the next, which is secondary spermatocyte, which is this one, it has come even down. And then from there, if you see the sperms, or sorry, spermatids, which are this one, they have come even further down. And then finally, they will be released in the lumen. So do you appreciate that from the top layer, as they are maturing, they are also descending down in towards the lumen, right? When I say down, I mean from the boundary towards the lumen, not actual down towards the gravity. Does it make sense, Hiram and Rabia?
Yes, sir.
So these cells are also moving, and who's helping them to move? Can they move on themselves by themselves? No, because they do not have locomotory, any thing to help them with movement. So the only cell that can help them move is the nurse cell, which is called Sertoli cell. So Sertoli cells throughout this journey is helping the spermatogonium and primary spermatocyte and secondary spermatocyte with nutrition. It is helping them with locomotion and protection. Okay, is it clear? Is that clear, everyone? Perfect.
Yes, sir.
Now, okay, now primary spermatocyte is the first cell that undergoes meiosis. So between this, you can see here they have mentioned here meiotic division 1. And we have already discussed that meiosis happens in 2 cycles. So the one cycle for meiosis includes 2 times division cycles. So meiotic, which is called meiosis 1. And as a result of meiosis 1, from one primary spermatocyte, 2 secondary spermatocytes will be formed. Can you see here? Yeah.
Yes, sir.
And then from each of these, so this is secondary spermatocyte. Now secondary spermatocyte, tell me, will it be haploid or diploid? Meiosis 1 has happened. Meiosis 2 is yet to be happen. So secondary spermatocytes will be haploid or diploid? Remember class 10th when you studied meiosis and mitosis, you studied in detail leptotene, zygotene, pectine, all these things, right? So there was a statement that called meiosis 1 is a reductional division and meiosis 2 mimics mitosis, right? Meiosis 2 is just like mitosis. It is equational. So it is meiosis 1 where the reduction happens, and then meiosis 2 is just like mitosis, only with the half the number of genome set. Okay, and it also makes sense because if you reduce 2 times, then the problem will be if you start with any number, let's say 46 human chromosomes, right? So meiosis 1 happened and there are 23 and 23 in each cell. So this is clearly reductional, right? Meiosis 1 is reductional. Now, if meiosis 2 is also reductional, then it will be 11.5 and 11.5, right? Can this happen? Can we have 11.5 chromosomes? No, right? With any whole number you start, if you reduce it 2 times, half every time, it will end up with the decimal, with a fraction, right? And which is not possible. We cannot have 11.5 chromosomes. What is 0.5 chromosome, right? So meiosis 1 is reductional, but what happens is this meiosis 2 is same. There will be 4 cells at the end, but each will have 23, 23, 23 and 23 chromosomes. So pay attention to this part here, which is meiosis 2. Do you realize that in meiosis 2, the mother cell and the daughter cell have the same number, no reduction? Because it is already a reduced number, which happened in meiosis 1, right? So that's why we say, write down this, it's very important. If these concepts have not been cleared in class 10th, this is the time to clear it. Meiosis 1 is reductional in nature. Meiosis 1 is reductional in nature, whereas meiosis 2 mimics mitosis. Meiosis 2 mimics means just like mitosis, behaves like mitosis. Meiosis 2 mimics mitosis and is equational in nature, so it remains equal. Is that part clear, Hiram and Rabia?
Yes, sir.
So if this is clear, tell me what will be the ploidy
Your
of
voice is
secondary
lagging.
spermatocyte, haploid or diploid? Sorry. Okay. Is it better now?
Yes,
Wait.
sir.
Is my voice better now?
Yes, sir. It's
Okay. My question is, if you have understood meiosis 1 and meiosis 2, tell me what will be the ploidy of secondary spermatocyte? They will be haploid or diploid?
haploid.
Haploid, yes, because meiosis 1 has happened, no? Do you understand, Iram? Between primary spermatocyte and secondary spermatocyte, meiosis 1 has happened, not meiosis 2. And meiosis 1 is reductional, it reduces. So from diploid, it will make secondary spermatocyte as haploid. So secondary spermatocyte is the first haploid cell type in the testes. Write down. Secondary spermatocyte is the first cell type which is haploid in spermatogenesis. So for the first time, reduction has happened in terms of genome, copy number, and it has happened at the level of primary spermatocyte to secondary spermatocyte conversion, where meiosis 1 takes place. Now the secondary spermatocyte still is not fully mature sperm, so it has to complete meiosis 2 in order to become spermatids. And when meiosis 2 finishes, from 2 secondary spermatocytes, you get 4 spermatids. Can you see that? And we call them
Yes, sir.
spermatids because they are not perfect fully sperm. They are still round, spherical in shape, and they will be haploid or diploid. What will they be, Iram? Spermatids will be haploid or diploid?
Haploids.
Haploids, right? Because once they are haploid in meiosis 1, they will remain the same after meiosis 2. Okay, haploid. So from secondary spermatocyte onwards, every cell is haploid. Secondary spermatocyte, spermatids, and mature sperms all are haploid. Now you see in this diagram there is a difference between spermatids and mature sperm. What is the difference? What can you pick out? Spermatids do not have tail, right? Can you see they are roundish, spherical? Whereas sperms need to have tail. Without tail, sperm is nothing, right? So this process, yes, so this process of maturation of spermatid into sperm is called spermiogenesis. Write down. Spermiogenesis. Spermiogenesis is specifically the name given to this one stage conversion between spermatid to mature sperm. Okay, spermatid to mature sperm. So which involves formation of tail and their structure getting more elongated from spherical. And this, for this process to happen, the most important role is played by Sertoli cells. Because you see in the diagram, I will clear the diagram a bit. Can you see this region where my cursor is hovering here? See all these heads.
Yes, sir.
These heads are, which is called spermatid cytoplasm. They are all embedded in the Sertoli cells, these yellow things, right? So their head is in the Sertoli cells and then they develop the tail. So they are kind of hanging into the lumen where their head is inside and their tail is formed in the lumen. And when the sperm head is fully mature, it forms acrosome on it. And we know what is acrosome, right? So, okay, we'll talk about sperm structure in 1 minute. But when the spermatid become mature sperm, okay, it has a fully grown tail and a neck and a head, and on the head there is acrosome, and then these detach from the Sertoli cells and fall in the lumen, and through the lumen they travel all the way back into the lobule. They are already in the lobule, so they travel all the way back into the retetestis, from the retetestis into the vasa efferentia, epididymis. There they mature and then go through vas deferens, and then they are ejaculated out. Make sense? So
Yes, sir.
spermiogenesis is clear, the maturation of sperm from spermatid, the growing of tail, acrosome, and neck is called spermiogenesis. And there is one more term in NCERT which is called spermiation. These two are different. So write down, define what is spermiation. So the question that can come in boards is, what is the difference between spermiogenesis and spermiation? So spermiation, write down, is the process of release of fully mature sperm, into the lumen, by the Sertoli cells. Okay. So there are 3 confusing terms: spermatogenesis, spermiogenesis, and spermiation. Okay. Spermatogenesis is the overall process of formation of sperm inside the testis that we studied just now, starting from primordial germ cell to mature sperm. Okay. Even spermatogenesis also includes sperms going into the epididymis and getting functionally maturation. That also includes in spermatogenesis. So the overall process that happens in the testes and epididymis is spermatogenesis. Spermiogenesis is that one step where spermatids develop into mature sperm. Is that clear? And then
Yes, sir.
spermiation is the final step where mature sperms are released into the lumen by the Sertoli cells so that they can go back into the retrotestes and forward into the epididymis. Is it clear, everyone? Rabia and Iram? No doubt in this? No doubt in any stage,
No,
which is
sir, no
haploid,
doubt.
which is diploid, which comes first, which comes late. Perfect. Very good. Now, quickly, the last thing for today, write down structure of sperm. Structure of spermatozoa. Now, just let me see if I. So the sperm has a head part. It has a neck part. Okay, and then it has a tail part. Now, the head part is covered with a cap-like structure at the top called acrosome. Okay, and inside is the nucleus, because its sperm has the nucleus, and in the nucleus it contains 23 chromosomes. The neck part is special because it is lined by series of mitochondria. Okay, and then finally the tail part. So it is divided into head, neck, and the tail. Is this clear? And this is the
Yes, sir.
nucleus of the sperm, and there will be cytoplasm in the cell. And okay, and in the neck there will be mitochondria. Each sperm has fixed number of around 55 to 70 mitochondria lined up in the neck. And these mitochondria power this cell, power the sperm to show the movement. Let me turn on my video to explain it to you. Am I audible clearly and also visible? No voice lagging, right?
No, sir, it's clear.
No. Yes. So imagine again, this finger has a sperm towards my, this is the head and this is the tail, which is free area. And at the neck junction are these mitochondria, because from the neck onwards, this needs to move vigorously, right? The tail needs to move and to propel the whole cell forward. So all the power comes from this neck. So because of this neck portion, this tail can move like this in a 3D motion. Okay. It propels like a 3D motion. Is it clear?
Yes, sir.
And whenever a sperm fertilizes an ovum, only the head enters inside the ovum. This is important, write down. Only the head enters the oocyte during fertilization. And why is that? Because the only thing which is required is chromosomes. Sperm is there to deliver. Sperm is a delivery agent just to run the fastest. Okay, the fastest the better. The one who will deliver the pizza first will get the prize. So it's like that. The first sperm who will deliver the nucleus first gets to fuse and make zygote. So only the head part, from this part onward, it's cut. So this goes inside and everything else, the neck and the tail, their job is done, so they don't enter inside the oocyte. Is that clear? Any doubt?
So where does
Yes.
the head goes? So sorry, where does the tail goes?
Nothing, it just stays there and then degrades.
Oh, okay.
It just stays. So so if this is the membrane of the oocyte and neck has penetrated, the head goes inside, the tail stays like this, and when fertilization happens, then this part will just degrade, nothing will happen. Okay, and for that matter, let me just tell you, only one sperm can enter each ovum. And what happens exactly at this time of fertilization is a beautiful biology. What prevents multiple sperms from entering? How biology decides that multiple sperms should not enter, otherwise there will be a problem. Because if with one female nucleus, two male nucleus fuses, it will be triploid structure. And a triploid human cannot survive. A triploid zygote will die and there will be miscarriage of the pregnancy. So it is prevented, only one sperm enters, and after that something happens that does not allow any other sperm to enter. This we will study in later part of this chapter when we'll come to pregnancy and fertilization. Okay.
So then how do twins form?
Yes, we will also study twins very interestingly, both in reproduction and genetics. So just hold on to your horses of the questions. I know it's very interesting. Twins also are of two types, identical twins and non-identical twins. So both have beautiful biology and different biology altogether. Okay, we'll talk about identical and non-identical twins as well. But this is it for today's class. The whole process is clear. Yeah. And I'm giving you homework to write down your homework for today. You will also be reminded later on, and please you have to submit also. I think you have to click pictures and submit it on Learnivio or something. Have they told you to keep submitting your homework assignments? Please do that. Okay, you are being assessed on that basis. So your homework for today is to write short note on the following: 1, Leydig cells; 2nd, Sertoli cells; and 3rd, spermatogenesis; and 4th, structure of sperm. Okay, just one paragraph. If you can make this diagram, very good, and then the submit. Okay, is it clear, Rabia? Aniram?
Yes, sir.
Okay, perfect. So if there's no question, no doubts, let's stop here and we'll